neurological aspect of voice production. As you just heard extensively, laryngeal function encompasses a number of behaviors, and one of them, most vital to us, is respiration, which includes not only breathing itself, but also coughing, throat clearing, sniffing, sneezing. Another important function is phonation, and this includes not only speech production, but also voice produced for laughing, crying, shrieking, speaking itself, and as well as singing. And finally, swallowing, where laryngeal function is also quite important. So, how is the central laryngeal control is organized to regulate these behaviors? All of them have something in common, but also have their own differences. So, it turns out that there is a hierarchical system in place to control our voice production. And the lowest level of this system is located in the brainstem. It's motor and sensory phonatory nuclei, as you see here. This system coordinates laryngeal, articulatory, and respiratory activities for innate type of vocalizations. For example, first crying of an infant, or vocalizations that are present in early childhood, as well as our own innate vocalizations when we produce something involuntarily. However, as children grow, or as we become more precise in our level of voice control, we engage higher levels of brain organization, and one of these is periaqueductal brain that is connected directly with brainstem nuclei and regulates the control of emotional vocalizations. Due to proper function of this subsystem, children are able to cry when there is no painful stimulus present, more like for seeking attention, and actors can mimic different emotional vocalizations on stage. This subsystem gets a number of other inputs from hypothalamus, midline thalamus, and so on. And importantly, from a region in the brain that's called cingulate cortex. Together, this subsystem is involved in vocalization initiation and control of voluntary emotional vocalizations. And finally, the highest level of this hierarchical organization involves laryngeal orofacial motor cortex with its output and input systems that help actually to control our speech production as an example of learned voluntary behavior. So I'm going to focus on this subsystem because we're going to talk about voluntary voice production for speech. So I will talk a little bit about the organization of the laryngeal motor cortex, the complexity of speech production control in terms of network organization and network interactions, as well as a whole brain functional connectome of speech control. So let's start with the organization of laryngeal motor cortex. Our early views on how and where the larynx is represented in the motor cortex come from studies in non-human primates, where laryngeal areas have been identified, laryngeal muscles, separative muscles, have been identified in the lower part of the precentral gyrus within the motor and premotor cortex. It was not until 1950s when Penfield and his colleagues mapped in detail different body representations within the precentral gyrus in the motor cortex. And among those, he mapped also something relevant to laryngeal motor control, laryngeal muscle representation. As you can notice here, there are different body regions represented on this slide. Hand, ring finger, neck, brow. However, in terms of speech control, there is this large area with the arrow to vocalization. So vocalization is not a body region, as you know. It's a behavior that encompasses both respiratory activity, laryngeal activity, and orofacial activity. So because Penfield and colleagues have not mapped in detail the laryngeal muscle representation, they rather showed a larger extent of a region that would be, in fact, responsible for all different aspects of voice production. And as you can see, that area also encompasses lips, jaw, tongue, and is positioned right above swallowing. So to further understand where and how this laryngeal motor cortex is situated and how it is involved in control of voice and other laryngeal behavior, several studies have been done in late 90s, early 2000s. And one of these study by our group where we looked into different laryngeal vocal behaviors in healthy individuals. They were asked to produce repetitive vowels. They were asked to cough and sneeze and also breathe voluntarily in the scanner. And here you see the paradigm of brain imaging study, fMRI study, where they were presented with a stimulus and they had to reproduce this task, and after that we collected their brain imaging data. So analyzing this data, we looked into how voice production may overlay with respiration. Given that respiration and breathing are vital for all our laryngeal functions. And here you see the inflated brain views from the side and from midline, where different colors represent different behaviors, like in blue is repetitive vowel production, activity associated with vowel production. In yellow is voluntary breathing, and in red it's overlap between these two behaviors. As you can see, there are a number of regions that are in red, and that makes sense since, again, we need our respiratory activity to produce voice for speech. But there are also regions in yellow or in blue that are responsible, these areas are responsible for control of vowel production or control of voluntary breathing separately. With a focus on the laryngeal area or speech area of the motor cortex, you can see that the activity is very similar between these two behaviors. However, it's much more pronounced during vowel production than breathing. As I mentioned, there are also some regions that are specifically active during vowel production, and in this case, it was periaqueductal region, which I mentioned in previous slides, is involved in control of voluntary voice production. Moving to other laryngeal behaviors, such as cough, we again see, as an example of forced expiration, you again see a lot of activity overlap between the two behaviors, but again, a region in frontomesencephalic junction that is representative of cough control. And finally, sniff, as an example of forced inspiration. Again, similar picture, but slightly different mapping of these regions that are involved in these behaviors. With, again, some regions in olfactory cortex and amygdala responsible for control of sniff. We have been discussing just during the break how our memories are formed during sniffing, and we can remember easily things that have happened decades ago just by smelling, having similar smells hit us, and these are the regions. Amygdala is especially involved in forming memory for sniff. So, putting this together, there are specific brain regions that are involved across different behaviors. And these areas, especially in the laryngeal sensory motor cortex, they are responsible for convergent control of these different laryngeal tasks. On the other hand, there are regions that are task-specific and divergently activated, and I already told you about these areas, just a reminder here on the slide. So these and several other studies looking into voice production as well as other laryngeal behaviors led later to intracranial mapping of laryngeal motor cortex in healthy humans. In humans who undergo neurosurgical intervention, but they don't have voice or speech problems. So, and here you can see the outline on the right where regions in black, I think you can move this, regions in black represent laryngeal motocortical representation, rather here, and this is more of a sensory area that is being also involved during voice production. These data were acquired on 3 human subjects during surgery and follow meta-analytical studies of existing fMRI literature during production of voice, syllable, and speech. Similarly map this representation in the laryngeal motor cortex using neuroimaging methodology. So, a little bit about structural organization of laryngeal motor cortex. For this very powerful methodology exists that's called diffusion-weighted imaging that measures diffusion of water molecules in tissue based on random walk of water molecules. As an example, if you take tissue, Kleenex tissue, and you drop ink on Kleenex tissue, you will see that it spreads so-called following Gaussian distribution isotropically. On the other hand, if you take a newspaper tissue and place the same drop of ink, you see that the diffusivity of these ink molecules shape elongated oval because of anisotropic distribution. Similar processes happen in the brain depending on white matter, myelinated and non-myelinated fiber distribution, and we can use these properties to track non-invasively white matter organization responsible for controlling specific behavior or for any general purposes. So diffusion-weighted imaging senses structure of the tissue by measuring water molecules displacement along a chosen path. And if that path of water molecule is affected by restrictions such as cellular material or other healthy or disease processes, the measured diffusion coefficient is reduced. If the tissue structure is oriented specifically, as I explained on the example of Kleenex and newspaper tissue, the path of water molecule also will reflect this orientation. So putting this together, we can generate these beautiful images, but they're also meaningful. Here you see 3D reconstruction of white matter tracts, and following the same principle, we can look into structural connectivity of laryngeal motor cortex by placing so-called seed, this green dot, in the region where we see peak of activity during vowel or sentence production. And from there on, you see the distribution of white matter pathways from that laryngeal motocortical area. So, putting this together, we can also see that different laryngeal behavior, despite the fact that they have different level of activity, they form very similar structural networks, which means that our laryngeal behaviors, no matter what they are, they have a unifying similar framework. On top of that, though, there is a selective activation that is produced or is required for the production of specific laryngeal behavior. Here you see in top panel syllable production or panel voluntary breathing, and the regions with red is where structural and functional connectivity overlap, and regions with yellow are the regions of only functional connectivity. Interestingly, you also see that voluntary breathing is more bilaterally distributed hemispheric behavior, and syllable production, similar to speech, which is a well-known fact, is more left hemispheric driven. There are specific also regions such as anteroparietal cortex and anterofrontal gyros that are responsible for driving this hemispheric laterality. So moving on to the complexity of speech production control, I want to talk a little bit how different networks interact with the laryngeal motocortical network and how they inform the control of these behaviors. There is a relatively complex system of speech and language production by Hickok and Popple back from 2007. And here you see that these regions in bluish, these are the ones that are responsible for speech motor output. And they tightly integrate and merge with other subsystems responsible for spatiotemporal analysis, lexical interface, phonological network, and so on. So breaking this down for the speech motor control, in this study we've placed seats for network tracking in 7 regions that were showing the highest level of activity, and this included laryngeal motor cortex, inferofrontal gyrus, also known as Broca's area, inferoparietal region, thalamus, putamen, and so on. And here you see it's being cut off. It says shared functional network during speech production. You see here how different networks merge with each other, and below you see the percentage of the overlap. So pay attention if you can see that there is a black dot around this area which has the highest overlap, and that's the laryngeal motor cortex. So these networks, all of these networks fully overlap in the laryngeal motocortical area, which is a logical way to explain that this is the final cortical motor output for our voluntary speech production. Similarly, you see resting state functional networks. This is when we don't speak, but these are resting state functional networks that are associated with speech production. And you see that there are some differences in addition to the overlap, 100% overlap of the networks in the laryngeal motocortex area. There are also areas again in inferoparietal cortex and cerebellum here that are quite empty during resting state, but they fill in and they get activated when we speak. And cerebellum is involved in regulating the tempo, for example, of speech production. So this is quite an important difference between resting state and speech production. So to summarize, the laryngeal motocortical network fully overlaps with all other speech-related networks and determines the extent of network-related interactions. And the convergence of network interactions in specialized brain regions is critical for speech, but not really for resting. And finally, whole brain functional conic dome of speech control. What is the connectome? Connectome is a large-scale network, and for speech production we need not only one region, like laryngeal motor cortex, but also its interaction with a number of brain areas, as I showed you on previous slides. Connectomes exist in everything. Networks between people, nervous organization in a worm, nervous organization in a human. So they exist everywhere. Here you see how networks can be integrated across different brain regions. You see some areas densely integrated, some areas less. So understanding these properties is important for different purposes. For example, figuring out the pathophysiology of some disorders. Networks are also segregated, where you can see that subpopulations of areas form these neural communities, and this is again important in our understanding of general organization, both in healthy individuals and patients with neurological voice and speech problems. And finally, there are specific brain regions that have higher influence on the network than others. And again, this is important to understand which regions we should be targeting for therapeutic interventions, for example. So let's look how these connectomes are distributed between resting and speech production. Both conditions share a number of brain regions within the sensory motor areas, and both cortical and subcortical sensory motor areas. However, they construct very different output, the connectivity with the rest of the brain when we go from resting to speaking. You see that some regions, like in purple, they get engaged only during speaking, but not resting, while the rest is kind of intermixed between the two behaviors. But resting and speaking are very different behaviors, so it is more logical to look into similar behaviors, for example, vowel production and speaking. Vowels being meaningless repetitions, motor repetitions, and speech being meaningful output, or most of the time, meaningful output. So here again you see there is this core network that is shared by vowel and speech controlling networks. But again, despite their similarities in their behavioral similarities, this core constructs very different output in terms of connectivity with the rest of the brain. So, to summarize, functional connectome of speech control is highly complex, and it involves the control of a multitude of neural processes that enables us to speak. There is selective activation of network components that depends on the complexity of laryngeal motor task production, going from rest to syllables to meaningful sentence production. Thank you for your attention. I'm happy to take questions if there is time. 1 minute, 19 seconds left.